Good morning to everybody. I am particularly happy because today we have as a special guest a friend and a great scientist. Is Noemi Roy. Noemi Roy holds degree in Bachelor of Medicine and Surgery and Bachelor of Science, and she's also a PhD. She has obtained a membership in the Royal College of Physicians of the UK and a fellowship in the Royal College of Pathologists. Currently, she serves as a consultant hematologist at the Oxford University Hospital NIH Foundation Trust and holds an honorary senior clinical lecturer position at the University of Oxford. She also serves as the Rare Inherited Anemia lead on the National Hemoglobinopathy panel. Her primary areas of interest are hemoglobinopathies and rare inherited anemias, particularly focusing on the genetic investigation on the inherited anemias using next generation sequencing approach, including whole genome sequencing. She has co-authored of numerous articles related to genetics and genomics of red cell defects and has made significant contribution to the identification of causative gene as exemplified in the case of CDIN1 as the second locus of CDI type 1 gene for the congenital dyserythropoietic anemia. In her role as a consultant of hematologist, she is dedicated to managing patients and coordinating best practice care in red cell disorders at the European level. She leads quality of life studies in sickle cell disease and explores innovative approaches to improve patients' compliance and pathway for high quality care. Noemi has had a long-standing collaboration with our group, contributing significantly to the development of guidelines of various topics relevant to the field of inherited anemias. Notably, she played a leading role in crafting guidelines on the use of NGS in the diagnosis of rare hereditary anemias, a topic she will present today. So, Noemi, please come to your position. The floor is yours.
Thank you.
And thank you.
Thank you very much, Professor Yolascon, and it's a real pleasure to be here today amongst you. And so thank you for the invitation. So I'm going to talk today about NGS for rare anemias and where do we go with variants of uncertain significance, VUSs. The talk is divided into three portions. The first is I'm going to go through the current NGS guidelines that Professor Yolascon just said that we published recently. Secondly, I'm going to explain how VUSs can be investigated. And thirdly, focus on culturing collaborations. So for the first part, these are the guidelines that we published a year ago. You'll see some of the names that will be very familiar here. And we did this jointly across the European networks. And it's a joint guideline between the British Society of Hematology and the European Hematology Association. And it's based on the fact that genetics has really evolved a lot from the technology side. So we can now do next generation sequencing, and we can now provide very precise molecular diagnostics. But what we wanted to get through with the guidelines is that you always need to start with the basics, and that is the patients. We need to start with a proper history and examination of the patients, and we will do all the usual blood tests, looking at full blood count, reticulocytes, hemolytic markers, looking at a blood film, and really we shouldn't be requesting NGS unless we've done all of these basics. In the guidelines, we've kind of talked about specialized tests like EMA dye binding, ectocytometry, red cell enzyme levels, and also looking at bone marrow pathology, and then finally looking at genetics. And we haven't in the guidelines said what order these should go in, and that's partly because it will depend on what country you're in and the funding situation, as well as what makes sense clinically. but certainly the method that is chosen by any lab should be based on local resources and on turnaround times. And also, it's important for the labs to be very clear with their referring clinicians about what this method is picking up, but most importantly, what it's not picking up. So if you have an area where you've got poor coverage of a gene, it needs to be clear to the clinicians that you haven't excluded mutations in that area. I thought I'd mention a little bit about how the genomic services are organized in the UK. So NHS England, who's our governing body for health, has divided England into these 7 different regions and said that each region can only have one genetic laboratory. And then each of those regions were invited to put a bid in to do non-malignant hematology genetic diagnoses. And so there's 4 regions that now provide this for the whole of England. And so Oxford is part of this central and south, and so we provide genetic diagnoses for this entire region. As part of this tendering process, NHS England has developed a national genomic testing directory, which means that if a clinician wants to order a genetic test, they have to look in the directory, see what condition is covered. So for example, in hematology, we've got our rare anemia panel. And then the 4 regions that are performing these tests all have the same panel with the same genes. So we've got about 100 genes that cover the usual things, the CDAs, Diamond-Blackfan anemia, the red cell enzyme and membrane deficiencies. We actually include hemoglobinopathies, but also things like sideroblastic anemia, methemoglobin, and a few of the iron metabolism. As part of our national test directory, we also say where in the pathway this can be requested and who can be requesting it, and what associated tests need to be done. The whole process has to be done in 12 weeks. So the turnaround time from getting the blood sample to providing the report takes 12 weeks. And when I give this talk to clinicians in England, they sometimes assume that it takes us 12 weeks to actually do all the work. And that's partly the truth because there's so many different steps in this process, but it's also to do with staffing levels and funding. So if we had better staffing levels, better funding, we could do it all in certainly 3 weeks and probably 1 week. But it is important for the clinical teams that request these tests that they understand that there's a lot of steps between sending the blood samples. We've got to extract the DNA and do the quality control. We prepare the library and barcoding. We can sequence. And the advantage of putting all of these tests together on the same platform is that it means you don't need to batch the samples together. So you can do some rare anemia panels and cardiac panels and neurology panels all together on the same sequencing run. And then we've got our quality control, read alignment, base mutation calling, and annotation and filtering. That's all happening in the central site. That's not involving anybody in hematology, but it's all a core service. Then the data is sent back to the clinical scientists, and this is where we've got clinical scientists that are specifically involved in hematology, and they do this analysis. We have an MDT, and then we write the report. So as part of these guidelines, we thought it was important to be clear about what criteria are essential for a lab to be able to offer clinical grade NGS. And that's because all of us who provide clinical grade NGS, we all started with just doing it in our scientific labs. And so it didn't have to be as stringent when you're trying to make a diagnosis if you're doing it for scientific reasons versus if you're doing it for a clinical reason. So obviously we've got to have consent for the DNA analysis. We've got to make sure we provide the phenotypic information. So I don't know how you do it here, but if we don't get the phenotypic information, we don't write a report. We need to be clear about the genes tested, and also copy number variants are not always picked up by all of these NGS methods. We filter the variants, usually between 1 and 5%. We need to be very clear about some variants that are not rare, but that have a modifying effect on our rare variants. And in red cell disorders, this includes the Lily and the lepra for alpha spectrin. We provide a false negative rate, and we have to fully validate the panel before we use it. So I'm going to talk a little bit more detail about the variant analysis that's happening by the clinical scientists. And this, certainly in the UK, we base it all on the American College of Medical Genetics guidelines. And that classifies variants from class 5 pathogenic, class 4 likely pathogenic, class 3 variants of uncertain significance, and then we've got our 2 benign ones. So for those of you who don't know or not aware of this, a class 5 variant is one that's been published and has lots of very good scientific demonstration that this variant and this gene causes this phenotype. And I've put here the seed-in that Professor Leo Lascon was just referring to. A likely pathogenic variant means you don't have to provide all the scientific justification because the location of the variant is such that it can only be pathogenic. So what do I mean by that? It could be a variant that's affecting the stop or the start codon. It could be a variant that's affecting the splice. So if it's a canonical splice variant at the beginning of the intron-exon boundary. Or it could be a frame shift variant where having one or two bases added or deleted means you've shifted the entire reading frame of your protein, and therefore it cannot be providing a sensible protein that will be functional. So these are what we call the likely pathogenic variants. But unfortunately, we still get about 40% of the variants fall under the variant of uncertain significance. So what should we do with these, and how should we report these in a report? So it's very clear that we include pathogenic and likely pathogenic variants in our report. We don't recommend reporting either intronic or splice variants that are non-canonical. So you can have some algorithms that predict whether a splice variant is likely, well, a variant is likely to affect splicing, but just having that prediction bioinformatically is not enough to be warranting reporting it. And we also don't report pathogenic variants that are not related to the condition. What I mean by that is you could be somebody investigated for Diamond-Blackfan anemia, and we find a pathogenic variant in pyruvate kinase, which means they will be a carrier for PKD, but we don't report that because that's not what they were tested for. So we thought it was also really important in these guidelines to say that labs should really be making a reasonable effort to share variants between each other. And we should also, if we're all doing the same kind of analysis, we should also be using the same transcript. For some cases, it may not matter which transcript you use, but for some genes, and one example is hexokinase, it makes a major difference if you're using the wrong transcript and you're reporting on the wrong transcript. I'm going to move on to the second part, which is what can we do about these VUSs and how can we investigate them? So first you try and predict if it's likely to be relevant or not. And the first question is, does the gene match the phenotype of the patient? Because if it doesn't, you don't need to worry about it, you can move on. You next need to look at the inheritance pattern. Is it what we're expecting for this condition? And then we can look at our predictive softwares. And our predictive softwares are based on two things. One is how much conservation there is between, for this amino acid, between the human, and if we go down as far as possible to the yeast. So the more highly conserved amino acid is, the more likely it's important that this amino acid remains as it is. And the second thing we look at is how similar or different the amino acid change is. So if you've got an amino acid that is been changed from one type, that's for example these polar uncharged side chain, to one that has a hydrophobic side chain, that's a big difference in the structure of the amino acid. So it's more likely that it will cause a big change in the conformation. But unfortunately, even if we use these predictive algorithms, that's not enough to tell us whether it's pathogenic or not. So what else can we do? Well, we can consider further studies, and that might include family studies. It might include some functional work, whether that's enzyme levels, EMA dye binding, ectocytometry, or CDA analysis. And these are the simplest forms of further work we can do. At the very most complex work we can do, we're talking about introducing that variant using CRISPR-Cas9 into a cell line to see if the phenotype occurs. But I'm going to show you some of these two simple cases, with two simple cases. So case one was a transfusion-dependent boy, and the thing about being transfusion-dependent means that you can't do any further analysis on the blood because you don't have any native blood to work on, so you can't do enzyme analysis. And both of his parents were healthy, and we found two variants in PKLR, which causes PK deficiency. One was clearly a class 5, and one was a class 3. So we're left thinking, this is looking exciting, it's probably PK deficiency, but we don't have enough information. The most important thing is to work out whether these two variants are in cis or in trans, because If one variant is coming from each parent, then it's possible that what we've classed a class 3 variant, a VUS, could be pathogenic and contributing to the phenotype. But if we do family studies and we find that one parent has both variants, then we can forget about it. We know that this class 3 is not disease causing. So in our case, we got the family samples and we found that the mother was carrying the class 3 variant. Now, what's really useful about red cell enzyme disorders is that if you are heterozygous, although you're completely healthy, measuring the amount of enzyme activity in a carrier, you get a normal level of activity, but it's at the lower end of normal. So here we did the PK analysis in both parents and found they both had low normal levels of PK. And therefore, we were now able to say, okay, this class 3 is actually a class 4, and we were able to upgrade the class 3 to class 4, and we were able to make a diagnosis for the family. Now, what do you do if you can't do that? If you don't have the parents available, or if they're living somewhere that they're not able to provide a fresh sample for analysis? Some laboratories would still report this. They would report the class 5 and then put a note about the fact that we found a class 3 and we're not sure that this is disease causing, but we would still want the clinicians to know, because that may be important for the clinicians to be able to make some decisions about how to look after the patient. This is the second case. So here we have two individuals, a boy and a girl. She presented. She presented in childhood, but we got to know her in adulthood. There's no consanguinity in this family. She presented age 4 with anemia and jaundice, and she'd required intermittent transfusions over her life. Had a splenectomy age 10, which reduced her transfusion requirements. The brother also had splenomegaly, but he hadn't had his spleen removed, and they were told as a family that they had hereditary spherocytosis. However, when we did the genetic analysis, we found 2 variants in SEC23B, and if we all know that CDA type 2, which is the gene that's coded by SEC23B, and hereditary spherocytosis often overlap clinically. So this seemed like it was a good idea that we should investigate this further. So one of the variants was a class 5, which was already published, and the other one was a class 3, which was predicted to affect splicing. We checked the brother. He had the same variant, so that's helpful, but it doesn't prove that this class 3 is pathogenic, so we had to go on to doing some further work. We did cDNA and electron microscopy. So I'm going to show you this variant. It's a point mutation here, 17 bases into the intron. So that's not, predictive software is not enough to tell us that it's pathogenic, because the only ones we can report are these 4, the 2 at the beginning of the intron and the 2 at the end of the exon. So we took some blood from the proband, we did CD34 extraction, and then we grew the cells in the lab, and we took some of the cells for electron microscopy and some for RNA work. And the RNA work we did was that this variant was predicted to cause exon skipping. So we expect the wild type to go from exon 13 to exon 14 to exon 15, and we expect the proband, if the variant is pathogenic, to skip exon 14. So it would go from exon 13 to exon 15. So it's very easy to do. We take the RNA, make cDNA, and then we just design primers to work on the cDNA to see if we get a product, if we try and find a product between end of exon 13 and beginning of exon 15. And that's what we found. So between our proband and our wild type, this is where our mutation is, and you can see that upstream of that, the sequence is the same, and then downstream the sequence is completely different because there's been exon skipping. We also wanted to look by electron microscopy, and we found that the erythroblasts, developing erythroblasts, had this sort of double layer membrane, which is typical of CDA type 2. So we had 2 lines of evidence to tell us that indeed what we found as a class 3 variant is actually a class 4. And so we now were able to give a diagnosis to this family. To show you just how useful these analyses are, we can look at the whole overall, what's the diagnostic rate. So we still get a fairly large proportion of patients in whom we don't find a diagnosis. And if you look at different publications, it varies between laboratories, but it's about sort of the diagnostic rate is between 40 and 50% in general. And when we do find a molecular diagnosis, in the majority of cases, they confirm a diagnosis that was already suspected clinically. So that's still very useful because we can then do genetic counseling for the rest of the family members. But importantly, in 10% of cases, we either get a completely different diagnosis or a completely new diagnosis. And that can have profound changes in treatment. What about the negative cases? Well, sometimes this might be because we've got the causative genes on our panel, but they're poorly covered, or actually it's a causative gene that's not been discovered yet, so it's not on the panel, or maybe we're not detecting the copy number variant, and also it could be a non-coding variant that we don't at this moment have enough scientific information to be able to detect. And so it's quite exciting to be moving towards whole genome sequencing and long-read sequencing to try and find some of these variants. But I still think that if we think about our 40% of variants of uncertain significance, if we can work together to try and improve the information we have about these, our diagnostic rate is already going to go up. So this brings me to my last part of the talk, which is about culturing collaborations. So as I said, we've put in our guideline that labs should make a reasonable effort to share variants with other labs analyzing the same genes, and also that we should be making sure we use the same transcripts. And we use the word reasonable effort because at the end of the day, every lab that's working on this is already very busy and very usually quite tight on time to meet the turnaround times. And a lot of this variant sharing involves extra time and effort and resources that very few of the diagnostic labs actually have. So we've done an initial comparison of the transcript between the 4 laboratories that provide NGS for rare anemias in Oxford, in the UK. And we did this comparison using 80 genes, and we found that for 66 of the genes, all the labs used the same transcripts, but for 14 of the genes, there were some differences. And actually, these differences depended on the lab. So it's not that one lab was using transcripts that no one was using. It was depending on the gene. Some labs were using one transcript, some labs were using another transcript. And then we also looked at the variants that we report. So initially we did the analysis between just Oxford and King's. So King's is in London. Oxford and King's were the first 2 labs that developed NGS for rare anemias, and therefore we had more experience. The new labs have only been doing it for about a year or 2. So we used these 2 different labs and found that there were 504 variants that were reported from both labs. I don't mean reported because some of these were benign, so they were just 504 variants that were detected by both labs. And you can see that in 2/3 they graded them as the same thing, the same ACMG class, and in 1/3 they graded it as different. But we have to think, what do we mean by different? So if you were grading it between a class 5 and a class 4, it doesn't make any difference clinically, so we should consider that essentially the same. If you're grading it a class 2 versus class 1, it makes absolutely no difference, so we should be considering that essentially the same. So what we mean by really different is either one lab called it class 4 or 5 and the other lab called it class 3, or a lab called it class 3, the other one called it benign, or even more strangely, one lab calls it class 4 or 5 and the other one calls it class 1 or 2. And those are the ones that we are most interested in investigating. And that represents 5% of the variants. You might think, well, why are we really interested between the labs that are calling it class 3 versus benign? Because that doesn't make any difference clinically, but actually it's very time saving. So if we know that a lab already has done some investigations and it's not a disease causing, and therefore it's definitely benign, then it can save time when the next lab sees it to just put it aside. And most importantly, when I showed you this extra work that we do, we don't want anyone doing all the extra work with electron microscopy and cDNA analysis if somebody else has already done it and shown it's not disease causing. So it should save money as well. So now that we've got the 4 labs doing it, we decided to do this comparison across the 4 labs. So King's has seen 4,979 unique variants, Oxford fewer, 1,700, and Sheffield and Manchester 300 or 400, and that's because they've only come on board in the last couple of years. And then we showed the number that were different between the 2 labs, and then we graded them as whether they were very different or clinically discordant. And then we've got in this chart so there's about 35 variants that were clinically significant differences between the different labs. And what you've got here is the Oxford is in blue, the King's is in orange, Sheffield is in green, and Manchester is in red. If you see a 0 here, it's because that lab didn't see the variant. So on the bottom, you've got the names of the genes. You've got aldolase, CDAN1, G6PD, lots of globin genes, piezo, PKLR, RAG, RPS10, RPS19, so those are diamond blackfan genes, SBDS, alpha spectrin, and beta spectrin. First thing I'm going to do is remove the globin genes. So hemoglobinopathies are always a special case, and there's other ways of looking at hemoglobinopathy variants that, because it's been going on for a long time, a lot of people don't follow the ACMG guidelines, so I'm going to not talk about those. So we're left with the rest of these, and actually some of these differences are very easy to resolve. So the ones I've circled, the aldolase, when we spoke to the lab in Sheffield, it turns out that they'd classified it as pathogenic because it was involved in a different non-hematological phenotype, and they didn't have much experience with this, so they didn't realize they should just not be looking at that. So that was easy. The codenin were very easy for us because we've done electron microscopy, so we were able to tell these other labs that they are pathogenic and they can increase it. We've got the piezo variants. King's has got an ectocytometer. They got one before we did in Oxford, and so they'd done ectocytometry, so they knew that these piezo variants were pathogenic. So some of them are very easy to resolve. But I thought I'll give you an example of one where it was slightly more, well, it's equally easy, but it really involves talking to each other. So this was a beta spectrin variant, which causes autosomal dominant hereditary spherocytosis. That's what SBTB does. And we had the following interpretation. So first of all, the phenotype is specific to the gene we've tested. That was fine. If we looked at our predictive information, remember I was telling you that we look at how well conserved the amino acid was, which in this case was deeply well conserved, and we look at how different the amino acid changes. So it was going from a polar uncharged amino acid to a basic amino acid. So that's the predictive software telling us it's probably pathogenic. We had EMA dye binding that was positive. The mother also had positive EMA dye binding and also had the variant, and the brother had negative EMA dye binding and didn't have the variant. But with these dominant disorders, it's very difficult. That's not enough information to tell you, to tip the balance and tell you that it's pathogenic. So that's why we had it as uncertain significance. But we spoke to the King's team, and they had another family. So here we've got another proband. They also looked at the mother of that proband, and that mother was also affected, and she also had the positive EMA dye binding, and she had the variant. And so now we have enough information that our statistics change, and they've done ectocytometry, that we could call it a pathogenic variant. So this brings me to the end of my presentation, which is some of you will remember that back in 2017, we started talking about how to collaborate on NGS. And in fact, I would say that the first output from that collaboration has been the guidelines that we've produced. But we now need to move on to the next bit, which is, can we start sharing variants across Europe? So there are 14 labs that participated, and we ran a sort of external quality assessment between each other, but I think we would now be able to start sharing variants. EuroBloodNet has, in theory, agreed to maybe sponsor a workshop on variant sharing, and so I really hope that certainly in 2024, one thing that we can do is actually sit down together around the table, and this would have to be something that would be face-to-face, but it would require a lot of preparation work. And I suspect we can't do it for all the genes in all the panels at the same time. I think we're just going to have to pick 1 or 2 genes and do the analysis beforehand about the differences, and then come together and share our data so that we can figure out whether one lab has more information and we can change the pathogenicity in the other labs. Ideally, we would all have one wonderful database where all of this would be uploaded and kept up, curated and everything. The worry is that would require a lot of money and a lot of effort, and I don't think we're going to achieve that very easily. And so I suggest that we start very pragmatically with 1 gene, spreadsheets, and take it from there. Last but not least, I want to thank everybody that's been involved. So this is the molecular genetics lab in Oxford. I've put the other labs that we collaborate with. So that's King's in London, Manchester University and Sheffield. And then in the research lab, these are all the people that are involved and working on helping us to determine on the molecular level whether some of these variants are pathogenic. Thank you very much.
Thank you, Noemi, for this wonderful presentation. It's absolutely very interesting because you touch a lot of problems related to the significance of the results. So this is a very important problem because the problem is we have to manage, in my opinion, is we have to manage a monogenic disease because it's, in my opinion, difficult to explain only in terms of one gene, one phenotype, because there are certainly the role of the entire genome. So also if you found two individuals from two different families with the same mutation, in my opinion, it's very difficult to say that it's absolutely the same phenotype. So this have a significance in the evaluation. But let's start with the questions to the presentation of Noemi. I will start with the people present in the room, but I ask to also for the people present in a remote manner. If they want, they could write the question, and after the first, I will come to the online question. Please write a lot of questions. Roberta?
Okay, thank you, Noemi. My first question is closer to what Professor Yalascon just said about the occurrence of multiple diagnoses, and in case you identify this situation, but the clinical suspicion is for only one of the two identified genotypes.
You mean two red cell diagnoses?
Yes, yes.
Yeah. So we've been very reluctant to call that. We haven't, there's not been any, I mean, I know you guys have published some really interesting cases, and we haven't found so many, but I think that's probably because we tend not to push that, because it's not always clear to us that how these genes interact and whether these genes interact, and especially because the particular, like piezo one, there's so many different there a lot of these genes are very pleomorphic, alpha spectrin, so we probably undercall our variants. But again, this is somewhere that I think we should put more data together, because it all boils down to statistics, and so if we put more data together, it will become clearer whether particular variants will interact across different genes.
Okay, and another question is always my same question. What about the mixed analysis of DNA-seq and RNA-seq to solve cases as you show for second?
So we don't have access to that, and there certainly are in the UK some initiatives to do that. Not specifically for red cell, but they tend to be national initiatives. So they will have a call to say, propose your disease, and we will do whole genome and RNA-seq. Our disease has never been chosen. Surprise, surprise. But yes, now I think also this brings up a real problem that I find in this field, which is that what we're interested in talking about here is incredibly important for the patients. But if you're a funder for research, you look at this proposal and you think, okay, it's a rare disease. We already can diagnose, say, 50% of them. You know, do we really want to put money into this this thing that's very translational. It's not going to tell us, I mean, it may, but it may not tell us very much interesting about science. All it's going to do is increase our diagnostic rate a little bit. And I find that really difficult to be at this interface between diagnostics and research, because certainly the NHS is not willing to pay any money to do these extra investigations, and then the research funders are not that interested in funding something that's feeding into what they consider should be part of the NHS. So you're kind of stuck in the middle. But I agree, this would be, if we could do this at a large scale for red cell disorders across Europe, I think we would have some really exciting findings.
Naomi, thank you for your presentation. In case of mutation in piezo-1 gene, do you perform ectacytometry as the second line of investigation or as a functional sign?
So, so as I said, there's only 2 ectacytometers in the UK. One is in King's and one is in Oxford. And in Oxford, we've not been given the right to set it up in the diagnostic lab because the lab is so tight for space and for money, etc. So we've had to get funding from a charity to buy the machine, and this was on the condition that nobody in the lab was going to use it except researchers. So we can't run the samples. And also the other thing is, geographically where the ectacytometer is not where the bloods come in. So the bloods will come in somewhere else. They will go to another place for DNA extraction. Then we get that it was a piezo mutation, too late to go back to ectacytometer. So what we do is we just keep it as a variant of uncertain significance. Then we request a fresh sample, and then we do ectacytometry. Yeah, it's not ideal, but yeah.
Okay. And moreover, I noted that you found splicing mutation piezo-1. What is the phenotype of this patient? Because we have some patients with splicing mutation, and we found that they are gain-of-function mutation, similar to missense variant in piezo-1.
So this is a patient that was seen at King's, and I haven't got any more information on them, but they have the ectacytometry is positive, yeah.
Okay.
But we could discuss it in more detail.
Okay.
That would be really interesting.
Okay.
Yeah.
Thank you.
I'm not much into blood diseases. My question is on the first part of the talk, where you showed the guidelines or the best practices. You say that you suggest to provide the rate of false negatives.
Yeah.
False negatives, right? So how do you actually calculate it?
So what we were doing is when we were using, we've changed the method that we use, and now we're doing more of an exome. But before, when we were doing the smaller panels, we had a lot of missing exons. So we would calculate what percentage of so we would only work on the published mutations. So for each of the genes, we knew the location of published mutations, and we would calculate how many of the published mutations were not included in the reads. So there were no reads that were covering them. So if there were 10 published mutations in one gene and one exon with 2 mutations had dropped out, we would say it's a 20% false negative rate. So it's a bit of an estimate.
Thank you.
Okay. And we have a question from remote. It's from Alessandro De Falco. Thank you for your clear presentation. Regarding the non-diagnostic category, what do you think about digenism, and what about the use of omics approach? The problem is that it's too little for me. I don't.
No, but I think it's basically what Roberta was asking.
Maybe the not diagnosed cases could be due to the not precisely defined clinical case.
Agree.
How can we overcome this problem?
Okay, so that's 3 questions.
Yeah.
But absolutely. So digenic inheritance, I agree, we don't know enough. I think what I think is that we need to do more basic science research and I think, using CRISPR-Cas9 to put variants into cell lines is a really great way to do it. So if we've got two different mutations in two different genes, we can put them in a cell line and see if it has a phenotype. The question is, what's your readout of the phenotype? So yes, I think we will get an answer to that when people who are working on it make more progress. The second one was, I think, yes, the omics approach I answered for Roberta. And the third one is this idea that actually, why do we get a negative diagnosis? Well, maybe it's not actually an inherited condition. So when we did whole genome sequencing, we did whole genome sequencing on 53 trios of anemias. So we've got the two parents, and it was usually children. And this was really at the beginning of whole genome sequencing in the UK. And this was all funded by the national government, and it took about 2 years before we got any results. It was really a long time. And because they were children, and Professor Yolascon, you're a pediatric hematologist, right? I'm an adult hematologist. So usually when people come to me, their phenotype is their phenotype. But pediatrics, my goodness, the phenotype can change so much. So after 2 years, sometimes we realize that this child has just got completely better. And even though they needed transfusions in the first months of life, now they're completely fine. They're not even anemic anymore. So then by the time we got the results, the phenotype had changed completely. So we need, and I think this is really important when we talk about doing omics. I mean, that's fantastic, but actually deep phenotyping is as important as omics. And the problem again is that we get to this sort of, who's going to put the time and the money into collecting all the clinical information. And I think that's where the RADIP, the database, the European database, can be really powerful if people can put all of the phenotypic information into the same database across Europe so that we can get more information. And in fact, for CDA1, I've been speaking to the people who do the American database, and I think we should work together with them, because the more information on phenotype we can have, the more clear it will be about, for CDA1, is it all one CDA1, or are there different types, or there's lots of uncharacterized CDAs. Maybe we'll get a better idea about the phenotype. So I think we need to work on that as much as on omics side of things.
Yeah. In your slides, I don't know if I see correctly, but when you are analyzing the same samples in Oxford and in King's, I believe that approximately 2% was a different.
So we graded the same variant differently. So one lab,
the evaluation.
Yeah. So one lab said this is disease causing, and the other lab said we don't know. And that's usually because they have more information, and that's why I think we should talk about our variants and what information we have to support it, because we can then probably increase the pathogenicity of all of these across,
To weight the variant, how do you consider the ethnicity of the proband? So it's for me, it's true English people or Pakistan people.
Yeah, it makes a big difference.
Or Chinese people, or black from Central Africa people. How do we evaluate the weight of the variant?
So usually you can tell when someone's of a different ethnicity because there's more variants, and that's just because we're comparing usually to the Caucasian population that's mostly been published. So we do our best, the clinical scientists do their best when they're looking at a variant to look at the ethnicity and see if in NOMAD or one of these big databases they can see what the variant allele frequency was in that population, because that's more important than across all the ethnicities. But yes, it is very important. I'll just say one more thing.
Who pay this analysis? Who pay
a
molecular analysis in England?
So the
The
NHS.
subject pay?
No. the NHS. So before we had NHS England that did the 7 regions, it was each hospital. So sometimes somebody would contact us and say, can you run the sample? And we say, yes, it's £500. And then they would say, oh, my hospital said no. And we would not do it. Now NHS England pays all the 4 labs, but they pay like a block contract. So they estimate how many samples you're going to get. But if you have, lots of negative samples, it's easy money. But if you have a lot of analysis to do...
Yes, this is absolutely true, because we have performed analysis for a lot of very different diseases. So one of the major problems in our work is the appropriateness of the sampling.
Yes.
Because I look at that if the analysis is from an hospital and it's completely free,
you get more samples.
The appropriateness go down in a very strict manner. But if you have to pay, and
it's more, yes.
And also you have to take care in a very stringent manner to the phenotype.
Yeah.
But if you not pay, it's not important.
So here our clinicians don't pay, the patients don't pay, the clinicians don't pay. If we receive a request form that's not filled in and that we don't have enough information, we don't do the test. Yeah, and I'm very strict about this. Yes. Now, the other thing that we've started doing is inviting our clinicians to the genetic MDT where we discuss the variants. Part of the problem for that is they assume if they get an email that they assume we found something exciting. So sometimes they appear on the Teams and then we say this is the case. We describe the case, no result, negative, and then they're like, why did you invite me for this? But actually, it's really useful. It's, genetic and genomic education of the clinicians who mostly don't understand a lot of these examples. So they can see how important the clinical information is. They hear us talking about what's a variant of uncertain significance, how are we weighting this? And I find that's really helpful. And a lot of the clinicians really enjoy the MDT because they're learning a lot.
Yeah. Oh, sorry. There is another question from it's from Professor G.B. Ferrero. Do you analyze negative cases with SNP array in order to look for germinal or acquired LOH?
So we don't, but sometimes we'll write it in the recommendations, we would say. So it's usually for children. So if it's a child that has something a bit more syndromic, we would recommend a CGH array. We don't do it ourselves.
From Professor Capasso.
Do you have a single hub for sequencing among these 7 regions?
No, there's several hubs, so each of them has their own
each
sequencing. Yeah. But I mean, the administrative side of things is crazy. So they've decided. So NHS England has decided all the DNA extraction has to happen for our region in Birmingham. So a sample is taken in Oxford. It goes up to Birmingham. It has DNA extraction, the sequencing, and then it comes back. I mean, it came back for library preparation, and it goes back up for sequencing. But I think this will change. We will be really centralizing more and more, and that's what they want. And really, Oxford is just going to be doing the analysis because that's where the expertise is.
Okay, thank you.
Yeah.
Please, Dr. Pinelli.
Thank you. Very interesting. I would like to make a comment that's like a take-home message for us that we are analyzing a mutation. About this inconsistency in interpretation. It's nice because when we see interpretation in clean bar, we tend to pretend that they are correct and sure.
Yeah.
But this is very clear. They are just another opinion. Either in the case are negative or positive. And just a comment for ourselves, just to remember this.
Yeah, and that's so we had the same thing with HGMD, which I don't know if you use here, but in the UK we were using HGMD a lot, and it's full of, SNPs that are described as pathogenic. So when I say it needs to be published to be a class 5, actually what happens is our clinical scientists, they find the publication and then they go through and they look at the data and they decide, is this really convincing or not? So yeah.
Okay, there are no questions. If not, I thank you, Noemi.
Thank you.
For coming here and for give us a very wonderful hour in this seminar. And thank you for your attention.
Thank you.